control of muscle strength. You know that we can control the strength of contraction. How do we do that? Well, let's take one step back before we answer that question. One is to put the different things that we've been talking about on the same time scale. So here in green is the action potential, the duration of an action potential in a skeletal muscle. So action potentials are very brief. So if for some reason the cell fired one action potential, it would look like this on this time scale. That one action potential propagating into the T-tubule, triggering the conformational change of the voltage sensor, triggering the conformational change of the calcium release channel, releasing calcium, would cause a rapid but not as rapid rise in cytoplasmic calcium, which would then get pumped back into the SR by the Surca. And so one action potential would produce a pulse of calcium that would have that time scale. And that pulse of calcium, binding to troponin, moving troponin out of the way, allowing the cross-bridge cycle, would generate a twitch. A short, only a few hundred milliseconds, twitch of the muscle. So, very brief action potential, longer pulse of calcium, longer force of twitch from one action potential. Now let's see if we summate, so here's two action potentials, two bursts of calcium, two twitches superimposed. 4 action potentials, 4 bursts of calcium, 4 twitches, etc. So this is temporal summation. And you can see that we reach sort of a steady level of contractile force because we reach a steady level of calcium. Calcium is now in sort of a steady state between being released and being pumped back up by Surca. So individual stimuli, if they come fast enough, generate a force. But slower stimuli generate less force than faster stimuli. Temporal summation. Let's not worry about the words here, but you do not have to worry about the frequencies here. The point is, higher frequencies lead to higher forces by temporal summation. That's mechanism number 1, by which the strength of a contraction is graded. The other mechanism, there are 2 mechanisms, the other mechanism is that our nervous system recruits fewer or more motor units. So if our nervous system says you're gonna need a lot of muscle to lift that thing, even before you try to lift it, it's saying recruit all the motor units you got so that you can use all the muscle fibers you got in order to make that contraction. So this is spatial summation. More motor units recruited to cause greater contractile force. And you've all experienced this, right? So if I, say I have, this is where you can see it. A bottle, right? So I happen to know, because I can sense it, that this bottle is full. But if I didn't know that, if say the bottle was just sitting here on a table, and I say, I think this bottle is empty. So I'm going to approach this empty bottle, and I'm only going to need a few motor units to lift this bottle. And then I try to lift this bottle with just a few motor units, and I realize, oh, this is heavier than I thought. My nervous system responds, recruits more motor units in order to do the contraction necessary to lift this bottle. I bet you've all experienced it where you've gone to pick up a box, and you think the box is full, and so you're ready to lift it with all your might, and the box is empty, and you do this funny jerky thing because you've used more motor units than you needed. Or the vice versa. You approach a box, you think the box is empty, I'm going to lift this, and it's like, whoa, I need more than that. Then you really get your motor units engaged, and you lift the box. That's using more motor units to lift the box. So it's using the nervous system, and you do both of these at the same time. So when I hold this bottle up, I am doing temporal summation, firing action potentials of the motor units that are contracting now, and I have used spatial summation. I've recruited enough motor units to lift this bottle. Not more than I need, not fewer than I need, to lift this bottle. And my proprioception is telling me, okay, I am successfully lifting this bottle. If this bottle were filled with lead, I would realize, okay, I gotta work harder. And proprioception would say, no, you're not lifting that you're not doing what you want to do. You need to recruit more muscle fibers in order to make the movement. If this was filled with lead shot or something, you can imagine how heavy this would be if it was filled with lead and not with water. That's how muscle fibers grade their strength. Temporal summation and spatial summation. Frequency of stimulation leads to temporal summation. Recruitment of more muscle fibers lead to spatial summation. Brittany, is everything okay? Okay. The final slide on this part of the talk is about muscle energetics. I don't want to dwell on this. It's an important part. All I want to say is that your body uses glycolysis, your muscles use glycolysis and oxidative phosphorylation to make the ATP necessary to do the cross-bridge cycle, necessary to cause the contraction. The ATP is also used by CIRCA to pump the calcium back into the SR after, when it's relaxing. So ATP is important here, both for contraction and for relaxation. I want to open up this story just a moment from what Self-Phys taught you, because Self-Phys taught you about glycolysis and the production of the oxidative production of ATP. And we went into something about lactic acid fermentation. We did some of this. What we did not cover in AMP, and which I'm not going to cover now either, is that protein, which is also a food energy source, and fat also feed into oxidative phosphorylation, but by different routes, right? Protein hydrolysis into amino acids, and fat hydrolysis into fatty acids do not go through glycolysis, but they do go through the Krebs cycle, and they do go through oxidative phosphorylation. And so our muscles use the sources available to it. Glucose, which you learned about, protein and fatty fats in addition. Okay, so that's one. I do want you to walk out of this course knowing that glycolysis from glucose is not the only source of energy. And a high-level biochemistry course would go through all the pathways necessary embedded in these areas. But I'm not going to do that. I also want to mention, and this will not be on a test, that we have ways to store ATP. We store ATP as ATP, and we store the high-energy phosphate in muscle, both in heart and in skeletal muscle, as creatine phosphate. So this is another high-energy phosphate. And we can use creatine phosphate to make ATP in short bursts. So creatine phosphate can donate its phosphate to ADP and make ATP. So we can make ATP by glycolysis, by oxidative phosphorylation, using food, and we can, for short term, regenerate ATP using a store of creatine phosphate. Questions about this energetics? Okay, so what I'm going to do now, we're not going to take a break. We're going to move right along to the next PowerPoint. And start to talk about the motor system, how the nervous system controls the muscles. So what we're talking about now, we're going to start to talk about the spinal cord and how it controls muscle. And then we'll talk about the brain, how it controls the spinal cord, which controls the muscle. That's what we're going to do for the next hour or so. Remember, we're talking about control of motor units. We're talking about how motor neurons control muscle. Now we're going to talk about how motor neurons are controlled. Let's do some anatomy. Cross-section of the spinal cord. Dorsal root ganglion. Sensor, so this is review. Dorsal root ganglion, sensory input in the dorsal horn. Now you should be remembering, okay, yeah, some of those are going to cross, the pain ones are going to cross over and go up to the brain via the anterolateral path, and the touch and pressure ones are going to stay ipsilateral, go in the ventral column, go to the brain, and then cross over. Right? You're working through the anatomy of these sensory systems in your brain, or at least you should for the exam. Now let's get back onto the motor. The cell bodies of motor neurons are in the ventral horn. So that's what I want you to write down for this slide. Cell bodies of motor neurons are in the ventral horn. Dorsal horn, ventral horn. This is anterior, that's posterior. This is right, this is left. Right, left, anterior, posterior, anterior horn or ventral horn. That's where the motor neuron cell bodies are. They send their axons out, and then they merge. And most of the nerves that we're talking about, the radial nerve and the ulnar nerve and the median nerve or the femoral nerve or the sciatic nerve are mixtures of sensory nerves and motor nerves because they merge here after the dorsal root ganglion. So here are the motor neuron cell bodies. Here's the output. You know where it's going. It's going to the neuromuscular junction, the synapse that we talked about just a few minutes ago. So here's an actual, so this is the cartoon, this is an actual stained section. Gray matter, white matter. So gray matter is pink, white matter is red in this staining. Dorsal horn, ventral horn. Dorsal root ganglion. Those are the words to know. The motor neurons in the gray matter of the spinal cord are mapped. They have a crude muscular map. So again, here's the dorsal horn, ventral horn, posterior, anterior. Distal muscles, like the hand muscles, are mapped laterally in the ventral horn. So laterally in the ventral horn are the distal muscles. And if we're talking about a cervical section, those are the muscles those are the motor neurons that control my fingers, lateral. If we're talking about a lumbar section, we're talking about, you can't see, but I'm wiggling my toes, right? The lateral muscles, the lateral part of the ventral horn controls the distal muscles, fingers and toes. The medial part of the ventral horn controls the proximal muscles, the muscles of the shoulder, the muscles of the hip. Okay, so a general mapish of the body in the ventral horn. Lateral, medial. There is also an anterior-posterior map. The extensors are more anterior, and the flexors, no, remember, the motor neurons that control extensors are more anterior, and the motor neurons, cell bodies that control flexors are more posterior. So there's a two-dimensional map. The motor neurons that control distal muscles are lateral, and the motor neurons that control proximal muscles are medial, and the motor neurons that control extensors are anterior, and the motor neurons that control flexors are posterior, all within the ventral horn. Which ventral horn depends on whether we're talking about arm in the cervical region or leg in the lumbar region. And in the thoracic, we're talking about the muscles that control the thorax, so there's a very small ventral horn here because we don't have very many muscles to control in our chest. The pectoralis major is actually is innervated both by lumbar and thoracic nerves. Okay, so now I've introduced the anatomy of the motor neurons that control the muscles. So we are here in this flow chart. We've introduced the motor neurons that control muscles, which are the effectors, and control movement. And there's a feedback loop because the proprioceptors in our muscles are giving feedback to the motor neurons and to the higher centers asking, is this movement what you wanted to do? Or are we off base? And so there's this feedback control. Your brain says move, you move, then you do this, error met. Did I move the way I wanted to move? And if not, let's make a correction. This whole system, well, the next step up from motor neurons are what's called central pattern generators. These are basic motor control units, and these are resident in the spinal cord. So the central pattern generators that we're going to be talking about are in the spinal cord, gray matter. They're not in the brain. So what's interesting is that a lot of our movements are stereotyped movements. Walking. Right? I'm initiating a program. My brain says walk, and then my spinal cord goes into a program and generates this program for walking. We have a motor program, and a lot of that programming is not in the brain, it's in the spinal cord. I know how to swim. So when I'm in the pool, which I wasn't able to do today, but I'm hoping to do someday soon, I know how to swim, and so I know how to control my arms in a swimming motion. I have a motor program that is resident in my cervical and my lumbar region that knows how to kick my legs and whatever this movement is, move my arms in order to swim, freestyle. It's a motor program. My brain says swim, and my spinal cord says, okay, let's go. Zoe.
Is this what muscle memory is? Or is that
Muscle
a different
memory
process?
is more, some muscle memory is stored in the spinal cord, but most muscle memory is stored in the cerebellum. Okay, so now we want to talk about these central pattern generators. And in order to do that, there are two reflexes that I use to build the concept of a central pattern generator. Because they introduce this idea of reciprocal control. This is a key concept. Reciprocal control. So let's talk about the knee-jerk reflex. You may have done this in a doctor's office. Can you see me online, people? Maybe turn up the lights. Oh, no, I think we're okay. So, the knee-jerk reflex. You know that if a doctor or a nurse or a physician's assistant or your brother hits your knee here at the patellar tendon, you tend to, I'm exaggerating, but it jerks the knee. That's the knee-jerk reflex. What it is it's a sharp stretch of the patellar tendon, which stretches the extensor muscles of the thigh. So it's a sharp stretch. Proprioceptors say, okay, we just stretched. And it triggers a contraction of the extensor muscles and a relaxation of the flexor muscles. Reciprocal control. In order to get a jerk, we need to have contraction of the extensors and relaxation of the flexors. Remember, this is extension of the knee, and this is flexion of the knee. So I'm hitting this muscle, hitting this tendon, and I'm causing extension. How does that work? How do we get reciprocal control? That's what's in this cartoon. Here we stretch the patellar tendon. The stretch receptors say, I just got stretched. They trigger that flow of information, action potentials, dorsal root ganglion, information, proprioceptive information coming into the dorsal horn. Some of that information is going to the brain, right? Going to the brain in the dorsal column, and your brain says, oh, I just got hit on the knee. But the brain does not control the reflex. The reflex is in the spinal cord, and it goes like this. In addition to going to the brain, there are synapses between these afferent nerves in the proprioception pathway and the motor nerves in the leg. And so green synapse here activates green to purple, activates the extensor muscle. Contraction of the extensor. It's a monosynaptic reflex. One synapse between the input and the output. Okay, good. How do we get relaxation of the antagonist muscle? There are inhibitory interneurons. You've seen this before for things like lateral inhibition. Well, this is not exactly lateral inhibition. This is inhibition of the antagonist muscle. Now, I'm using language that I don't think I've used before. Flexors and extensors are antagonists. They work against each other. When I flex my flexor, I'm stretching my extensor. If I try to contract both my flexor and my extensor at the same time, I could do it, but it's isometric. I'm not moving either of them because they're antagonists. Right now, you might be able to see. I'm contracting both my extensor, my triceps, and my biceps. Now I'm contracting only my biceps and relaxing my triceps so it can be stretched. The reflex does the same thing without even thinking about it. Input, excitation of the extensor, excitation of the inhibitory neuron, inhibition of the motor neuron that controls the flexor. So, automatic stimulation, your brain doesn't do this. If we were to numb this neuron here, you wouldn't feel the strike, but you would still do the knee jerk. If you are unconscious and your brain doesn't, is not sensing anything, you will still do the knee jerk reflex because it doesn't require the brain. It only requires a functional set of synapses in the gray matter of the spinal cord. That's what a reflex is. And all these reflexes are reciprocal, contracting one muscle and reciprocally relaxing the antagonist muscle. Okay, so we haven't built, I'm trying to get to movement. We're not there yet. Let's look at another reflex. It's slightly more complicated. This is the pain reflex. So you know that if you accidentally step on a tack, you withdraw your leg even before you know it. You put your hand on a hot stove, and your hand is off the stove even before it hurts. That's the pain withdrawal reflex. How do we do that? Okay, let's walk through it. Step on a tack. Pain neuron. Pain neuron goes up the leg. Pain axon. This axon of a pain sensing neuron goes up the leg. Let's say in the femoral nerve, dorsal root ganglion, in the, say, the lumbar region of the spine, enters the dorsal horn. Synapses on this, now we're doing sensory. You sense the pain because there's neurons that receive this input at this synapse, cross over, and go to the brain in the anterolateral column. That's a review of sensation. Now let's look at all this other stuff. The sensory neuron, the pain neuron, in addition to synapsing on the neuron that goes to the brain, synapses on motor neurons that innervate the flexors of the side that got hurt. So there is a mono or a disynaptic excitation of the flexor on the ipsilateral side, and through an inhibitory interneuron, activation of the inhibitory interneuron, inhibition of the ipsilateral extensor. This pain, I step on a tack, and I automatically flex my flexor, contract my flexors, and relax my extensors. But we are sophisticated. We know that if I'm standing and I automatically if I step on a tack and I do this, and I don't do anything on the contralateral side, I'm going to fall over. I need to not fall over. And so we have developed a reciprocal. So here's reciprocal on the ipsilateral side, and we've worked out a reciprocal innervation on the contralateral side. So excitation of the sensory pain neuron, excites the contralateral extensors, and inhibits through the inhibitory interneuron the contralateral flexors. So we may not be aware of it, but when I step on a tack and I flex the leg that got hurt, I am automatically extending the leg that didn't get hurt in order to not fall over.
Bob,
Yeah.
I'm not super sure of a scenario where this would be like realistic or something you'd want, but is it possible to resist that automatic response?
Oh yeah. So there are higher centers that can overpower this. We'll talk about higher control in a minute. You can override your reflexes, but it's hard, right? So if you know, say you've got the reflex is you put your hand on a stove and your reflex is to withdraw your hand. And in fact, you may not be aware of it, but if you withdraw your hand from a stove, there's a reflex extension of the contralateral arm in order to stay balanced. You can see that's how I'm starting to get a swimming motion. Reciprocal movement. But you know that if oh my God, there's a fire on the stove. I need to take that pan off the stove. I need to do it now. You can override the reflex withdrawal and say, okay, I'm going to get burned, but I'm going to have to lift that pan and take it off the stove. So your brain can override the pain reflex if necessary. Did that answer your question, Vincent?
Yes, there you go.
Okay. So now we're starting to see locomotion, reciprocal control, contracting flexors and relaxing extensors on one side, and contracting extensors and relaxing flexors on the contralateral side. And that's what we do when we move. We have a program. It's so automatic that I have a hard time even talking about it. Contract my flexors, and extend my extensors on the other side. Extend here, flex, extend here, flex here. You see what I'm doing? I'm alternating. Flexing and extending as part of locomotion. We evolved from simple creatures. So this is a vertebrate. It's a lamprey. Lampreys have this beautiful swimming motion. They also have reciprocal, alternating control, right? I don't want to go in, we don't need to learn about the lamprey movement, but movement of a fish or an eel or a lamprey, this contraction on one side, relaxation on the other, alternating with contraction somewhere else and relaxation somewhere else. That alternating is what generates this movement. The alternating of flexing and contracting, flexing and extending, is what allows me to walk. We actually there's different levels. So you may know that if I don't think about it too much, my arms are going to swing in the opposite movement of my legs. I can overpower it if I think about it. So I'm trying not to think about it. But, right? I'm walking, and I think about it, I screw it up. But you may see that when I step forward with one leg, I'm screwing it up because I'm thinking about it. But I already did. My arms are moving in the opposite pattern of my legs. We evolved from, hominins that were both walking and climbing. And so there is control. There are control units, there's rhythm generators in the ventral horn that are communicating with rhythm generators in other parts of the other parts of the spinal cord. You do not have to know anything from this slide, other than to know that there is communication between one level of the spinal cord and another level of the spinal cord, generating these complex motor programs. Your brain tells the motor program, go, and then the motor program runs. When you learn to walk, you are learning how to take all this input of balance and move stuff from your brain, and you're learning. I have a grandson who is a toddler now, and he just recently learned how to walk. And he's so proud of himself because it's hard. It's hard to get up on your hind legs and walk. And that's a program that he has learned. His brain has helped his synapses in his spinal cord learn the motor program necessary to integrate information from his proprioceptors and information from his inner ear in order to maintain balance and generate a program to, he walks like this. He's still learning how to walk smoothly. But it's hard learning. But it's in the spinal cord, which is interesting. There's a site here, which we're not going to show here because the quality of this image is poor. And it's an old video of a cat walking on a treadmill. But the tragic thing about this cat is it has no brain. It's been, the brain, the frontal cortex, including the motor cortex, has been cut away. It still has a cerebellum. You'll see, if you watch it, you'll see this, the cat has to be supported. It can't hold its weight up. But it can generate the movement of walking and trotting and running, depending on the speed of the treadmill. Because those programs of walking, trotting, and galloping are resident in the spinal cord of this animal that has no more brain connection. So we've learned about, where are we now? We've learned about muscles. We've learned about proprioception that gives feedback for balance and control. We've learned about central pattern generators in the spinal cord that control the motor neurons, that control the muscles, to generate stereotyped movement like walking, or running, or swimming, or dancing. And now we're going to talk about how the brain controls the spinal cord. Okay, you with me? In the precentral gyrus, so this is front, back, here's the central sulcus, here's the somatosensory cortex, which you learned about a week or so ago. Now we are in the frontal lobe because we are in the precentral gyrus. We're in the most posterior gyrus of the frontal lobe, the precentral gyrus. It's the primary motor cortex. And it has a map of the body that is quite similar to the map of the body in the somatosensory cortex. Leg and toes are medial, face and mouth are lateral. Hands and arms are superior lateral. Like this map of the body should look pretty similar to the map of the body's sensory input in the postcentral gyrus. Same idea. Lots of cortex devoted to fine control of our facial muscles. We've evolved to be able to control our mouth for language and the rest of our face for communication. We communicate with our eyes and with the muscles of our face and with our mouth. We are good at communication is one of the things that makes humans special, not unique, but special, because, and we use a large part of our motor cortex to control, fine control of those muscles. We are good at manipulating things with our hands. Lots of cortex devoted to the fine control of our hands. Not so much our arms and our legs, that's pretty basic, right? That's just motor control, movement, swimming, dancing, walking, running. We're pretty good at controlling our toes and our feet. It's important for walking. A motor map. The neurons of the motor cortex. So this is frontal lobe in cross section. I could play a game with you about naming things. Maybe we will before the end of the day. Longitudinal fissure. Cingulate gyrus. That's not what we're focusing on. We're focusing on the motor cortex. This is where the gray matter is. This is where the neuron cell bodies and dendrites are. The axons from those motor neurons, and sorry, they're not motor neurons yet. The axons from the cortical neurons of the motor system form this large amount of white matter and form the internal capsule. That's a name that you learned previously when we were learning coronal neuroanatomy. So the internal capsule, white matter, is formed from the axons of the motor cortex. Motor cortex, internal capsule, and it's going to form white matter paths that are going to go down the spinal cord. Let's skip the next slide. It's just another pretty picture of that. Here is a picture that you've seen before. Now I can talk about it in its entirety. What do we call this section right here? I'm standing in front of it. What do we call this section? Anybody? Dorsal column. What information is transmitted in the dorsal column? Is it sensory or motor? Sensory is blue. Sensory information, what kind of somatosensory information is carried in the dorsal column? Proprioception, touch, pressure. Ipsilateral or contralateral? Ipsilateral. Here is the anterolateral column. Pain and temperature information. Contralateral. Here are the spinocerebellar tracts. Spinocerebellar tracts. More proprioception information going to the cerebellum, some of which is ipsilateral and some of which is contralateral. We're not going to get into the details of which is which. Ipsilateral and contralateral proprioception. Ipsilateral proprioception, touch pressure information. Contralateral pain temperature information. Okay, just a review of the main blue pathways. Now the red are the motor pathways that are descending. Motor pathways from the brain going to make synapses in the ventral horn of the appropriate level in order to cause movement. So here's the new information. The main motor pathway is the so-called lateral corticospinal tract, which is the main part of the pyramidal tract. You can use either name, either pyramidal or corticospinal. That's this red area here. Now you're going to ask, is it ipsilateral or contralateral? And I wish I could tell you it was easy, but it's not. 90% but that's a number I do want you to know. 90% of the fibers in the lateral corticospinal tract are contralateral. They cross over in the medulla. So there's a crossing over. Remember, so this is one side, crossing over, and then here's the other side, crossing over. So upstream of this is the motor cortex. Upstream, the internal capsule. Here's the white matter in the upper medulla. And then in the lower medulla, it crosses over into the spinal cord and forms the lateral corticospinal tract. And 90% of the corticospinal tract is contralateral. 2% of the lateral corticospinal tract is ipsilateral. You don't have to memorize that number. 8% of the fibers aren't in the lateral, they're in the anterior corticospinal tract. You don't have to memorize that number. What I do want you to know is that 90% of the output of the brain, of the cortical motor, the motor cortex, forms the contralateral lateral corticospinal tract. So 90% of the axons in this area are from the contralateral part of the brain controlling, so this is the right side, so these neurons are going to control the right side muscles, right? They're going to make synapses here and control the right muscles, but the information is coming from the left cortex, the left hemisphere. Contralateral, 90%. 2% are ipsilateral, and 8% are in this other part right here. This could be on the exam. I could ask you to label a picture of the spinal cord. Meg, was that your question?
Yeah.
Okay.
So we could say, like, even though we don't need to know the specific numbers, we need to know the other two pathways.
Yeah. So I'm asking. So the main pathways that I'm asking you to know about are the dorsal column, the anterior dorsal columns, anterolateral columns, spinocerebellar tracts, pyramidal or lateral corticospinal tracts. All these little things down here you don't have to know about. Maybe for the future know that they exist, but for this test, no. Okay, so let's take a short break now. We're pretty close to done, but I want to get into the brain. So I want you to have a fresh brain to talk about the brain and the brain's control of the motor system. So we've talked about these cells, and we've talked, in the motor cortex, and we've talked about how those cells make those axons make the internal capsule. Now we're going to talk about some subcortical regions of the brain that are essential for movement. Let's take a 5-minute break before we do that.